Alternative pathway of complement activation. Complement components in our serum can be activated via three pathways: classical pathway, alternative pathway, and MBL pathway or mannose binding lectin. Today we are discussing with the alternative pathway of complement activation. As I discussed before, the only difference between all these three pathways is in the initial event. Ultimately, all this pathway leads to the formation of MAC complex and cell lysis. Okay. So, what are the key difference between classical pathway and alternative pathway? In classical pathway, we know that the pathway is once the antigen antibody complex is formed. Whenever a foreign substance enters our body, it is recognized by the antibody and form an antigen antibody complex. Once this antigen antibody complex is formed, that can further activate the components, like first C1 will come, then C4 get activated, then C2 get activated. So there is an order for that in classical pathway. So this antigen antibody complex initiate the classical pathway. But in case of alternative pathway, it is antibody independent. Alternative pathway is antibody independent. Okay, so why it is called antibody independent? Because once the antigen enters our body, the components of the antigen, certain surface constituents of the antigen, can directly activate the alternative pathway. No need of the formation of antibody or antigen antibody complex. So antibody formation is not essential. Formation of antigen plus antibody complex is also not required. Instead of that, antigen directly activate our alternative pathway. Okay, antigen can directly activate the alternative pathway. This is the major difference. Okay, so how this antigen directly activate alternative pathway? Before that, what are the major components of alternative pathway? Almost all the serum complement factor which participating in classical pathway, they are also are a part of this alternative pathway, but certain components like complement factor C1, C2, C4, that and all is not required for alternative pathway. Instead of that, certain other factors plays the key role, like factors, the complement factor C3, factor B, factor D, as well as one more factor called properdin. So these four are the major components participating, serum components participating in the alternative pathway. As I discussed before, in alternative pathway, the antigen's surface constituents can directly activate the complement. Once the antigen enters our body, whether it is a gram-negative bacteria or gram-positive bacteria or viruses, whatever it is, once the antigen enters our body, we know that the antigen's surface, like its cell wall, carry certain unique kind of constituents in it. Okay, so this constituents may not be present in our system. So for the very first time, they are introduced or they are exposed to our host system. So our host system recognizes all this antigen as foreign substances. Okay, they are they recognizes this surface constituents of the pathogen as foreign substances, or they recognizes that as the antigen. Okay, so they can find out that our body can identify that it is not ours, it is non-self components. So that things has to be eliminated from our body. This is the basic principle. Okay, so this antigens can directly activate the cleavage of one of the serum component called C3. Okay, so this antigen directly activate the cleavage of C3. Okay, in classical pathway we know that C3 is cleaved by C3 convertase enzyme. C3 is cleaved by C3 convertase enzyme and leads to the generation of certain components like C3A plus C3B. Okay, so the C3A is an inflammatory molecule which diffuses away, C3B participating in the classical cascade of complement activation. So the C3 convertase enzyme is essential in classical pathway, but in case of alternative pathway, the antigen directly enhances the spontaneous hydrolysis of the C3 complement factor. So the C3 will be cleaved and generate C3A and C3B. Okay, now the C3A as like normal diffuses away, they have inflammatory function, whereas C3B inserted into the surface of the pathogen. Okay, not only pathogen, they can also get inserted into the surface of our own cells also. Okay, this binding of C3B is not very highly specific and all. This C3B generated can bind to both pathogen surface as well as our own cell surface also. But we know that in mammalian cell surface, our own cell surface, which carry high amount of sialic acid in it, right? Mammalian cell surface carry high amount of sialic acid. So our own cells, if they are get inserted with the C3B also, no problem, that won't destroy our own cells, because the sialic acid will inactivate the bound C3B. But in bacteria and viruses and other pathogens, this level of the sialic acid is very minimum, very less. So it cannot inactivate the inserted C3B. So the C3B remain active there. So since this bacterial surface carry low amount of sialic acid, what happens? The C3B remain firmly attached to the surface and it can activate or execute the alternative complement activation pathway. How means? Once the C3B binds here, what happens means it exposes the binding site for the second factor, that is factor B. Once factor B binds here, this interaction is stabilized by a magnesium ion and it exposes the binding site for another factor, that is factor D. Once factor D associate with this complex, factor D enhances the cleavage of factor B, and then the factor B cleaved and generate two fragments, one smaller fragment called BA and one larger fragment called BB. So the smaller fragment diffuses away, but the larger fragment come and binds to this bacterial surface along with the C3B. Okay. So now we can write, instead of factor B, now we can write here BB. So the smaller factor B is cleaved and BA is released out, but BB remain attached to the surface of the bacteria. So at the same time, factor D, now you can see, right, it performed its function. So what happened? Factor D also diffuses away. Okay, now the complex we can read it as What is the complex now? C3b BB complex, right? So the C3b BB complex in alternative pathway, it is known as C3 convertase. Now you can go back and recollect in classical pathway the C4 the C3 convertase is named as C4b2A, but in alternative pathway the C3 convertase is formed later. In initial stages it is not required and it is C3b. So once the C3 convertase formed, what happens? The C3 convertase can cleave the freely circulating many C3s. As I discussed before, one C3 convertase can cleave thousands of freely circulating C3 complement into C3A and C3B. So what happens? The C3A is having inflammatory role, it will diffuses away, whereas the C3B, generated C3B come and binds to the surface of the bacterial cell. So many C3B get inserted into the surface of the bacterial cells, and some of the C3B again form complex with the existing C3 convertase. Okay, some of the C3B form complex with the existing C3B, so existing C3 convertase. So now what is this complex called? Just read the complex. Okay, so now the complex is called C3B BB, C3B actually, but we won't write C again, so 3B. So the form a new complex C3B BB 3B. Okay, this complex is known as C5 convertase. We know the function of C5 convertase. It will cleave the circulating C5 complement into C5A and C5B. C5A is having inflammatory role, it diffuses away. C5B, what happens? The C5B attaches to the surfaces of the membrane. So consider this as the C5B. So many C5B get inserted to the membrane. Once C5B is attached, it exposes the binding site for other complement factor like C6, C7 and C8. Okay. Once the C6, C7, C8 assemble along with C5B, what happens? It exposes the binding site, or it helps in the oligomerization of another complement factor that mainly helps in the formation of MAC complex called C9. So now C9 oligomerizes around this C5B 6, 7, 8 complex and form a pore like structure. Okay, a hollow pore like structure. So this hollow pore, through this hollow pore, but what happens? All the bacterial ions and other substituents diffuses away, and finally that leads to the lysis of the bacteria. Okay, so the ultimate event is same in classical pathway, alternative pathway as well as in MPL pathway. So that's all for today. Thank you.